Great, thanks, F. All right, let's get started. So today's presentation is entitled Southern African Population Structure, an Opportunity to Expand Genomics Research Worldwide. The structure of today's webinar, I'm going to chat very briefly about who I am and what I do, and then discuss how this research benefits worldwide populations. So who am I? I'm a postdoctoral fellow currently at Stellenbosch University. I started my career at UCT, and I did a BSc in molecular and cellular biology in 2010. I then moved to Stellenbosch in 2013 to do my BSc Honours in Human Genetics. I started my master's and was upgraded to a PhD, and I graduated with my PhD in 2017, and I started my postdoctoral fellowship that year, and ever since 2017 I've been in the same lab, same division, same department. I love it so much. I can't seem to leave. So in terms of the structure, so I am a fellow in the Department of Biomedical Sciences, in the Division of Molecular Biology and Human Genetics, and within that division, you obviously have the molecular biology side, and in the human genetics, the sections that I'm involved in is TB host genetics, clinical genetics, bioinformatics, and then I'm affiliated as an associate member to the Center for Bioinformatics and Computational Biology at Stellenbosch University. So what do? I do a little bit of everything, jack of all trades. I started off my career with population genetics and demographic history analyses of Southern African populations. I linked that up with infectious disease risk, particularly tuberculosis. The population genetics and demographic history analyses tie into looking at other clinical diseases, some Mendelian diseases, and then finally I'll chat a bit about my newfound love in bioethics and ethical, legal, and social implications surrounding genomic databases and genomic research in general. Okay, so let's get started on population genetics. So I think some of you will be familiar with this diagram from Desiree Peterson's paper in 2013, detailing the history of Southern Africa. As you know, the various migrations that entered Southern Africa, starting off with the first Southern Bantu-speaking population's arrival in South Africa, settlement in the Eastern Cape. You have the European settlers that arrived in the 17th century, introduction of the slave trade and settler-slave marriage. There were various epidemics. In the 18th century, smallpox, there's been documents of tuberculosis and other pathogens present that heightened the mortality rate. Slavery was abolished in the mid-19th century. And all of these events have shaped the genetic diversity that we see in Southern Africa today. Now this map is just a depiction of the variety of different populations, specifically Khoisan in Southern Africa. And one or two of the populations that I'm particularly interested in is the Nama in the Richtersveld, so northern South Africa, and the Komani in the Greater Uppington region. So because of their shared history, one of the initial questions that I was tasked to answer was, is there fine-scale population structure between these two populations? And I set off on this journey, coming from not knowing what a terminal was, not being trained in Bioinformatics, and I started off looking at global ancestry proportions and estimation. So in this plot, we use a number of different data sets to look at fine scale population structure. This work was done in collaboration with Dr. Brenna Hen. So if you look at the top panel, you'll see all the different populations that we used, as well as zooming in to the two populations that we are interested in here to answer the fine scale population structure question, and that is the Kumani and Nama. So if you're not familiar with admixture plots, every vertical bar is an individual. Each color is reminiscent of that particular ancestral population. So if you look at the French, the red color is reminiscent of French ancestry. The dark blue from in the middle from the Yoruba is reminiscent of West African ancestry. So if you use the same principle for all the different colors, you'll see that the Kumani and Nama look relatively similar. So we continued with this and we did principal components analysis, and you can see here that the Kumani and Nama do form somewhat distinct spatially separated groups, although there is some overlap. And one of the conclusions that we made was that at that time we needed more data to definitively show fine scale population structure, although there was it was suggestive based on this analysis. So what about genetic diversity of other populations in Southern Africa? Although I'm particularly interested in Khoisan, they have founded quite a few other admixed populations. So you'll see on the left, you'll have the Kumani again. The colors here are different. But what I would just like to point out is the extent of admixture that you'll find, particularly in the populations labeled colored NC, that's South African colored population from the Northern Cape, and the next population is the right is from the Eastern Cape, and SSE is from Western Cape, and then you have your District 6. So you can see there that there are varying levels of admixture, and they're quite heterogeneous. In this admixture plot, you also have the COSI, the Bantu-speaking South African population, as well as other African populations here. So one of the conclusions from this analysis, if you plot the genetic diversity or ancestral proportion based on geographical location, you can see some very interesting patterns coming out. So we identified 5 distinct ancestry patterns, first of being Circum-Kalahari Khoisan ancestry, followed by Central Kalahari, Northern Kalahari, Northwestern Savannah, and then we found the Eastern Bantu-speaking ancestry coming down, reminiscent of the migration. So we also, in terms of ancestry estimation, we also did not only global ancestry, but we did local ancestry. Dr. Alicia Martin assisted with this, and we local ancestry in essence estimates the ancestral origin of each chromosomal segment on the maternal and paternal copy of chromosomes. You can use local ancestry estimation for migration modeling, which you can see in the B panel of this figure, and you can use that to estimate the time since the admixture event. So all of this resulted in a publication in Genetics, my very first publication, so it was very exciting. But as with most scientific fields, you have knowledge gaps and room for improvement, which most of us are aware of. Is a lack of genetic data on African populations, tools and computational algorithms that are not suited to admixed populations or even African populations, since we have such a unique genetic diversity. So one of the students that I co-supervised recently published a review identifying some of these knowledge gaps. and we hope to begin addressing a number of these. One of the ways in which we would like to do that, particularly when it comes to genetic data, is I've recently established a South African human genetics database, which we just got ethical approval for. It's composed of genetic data generated as part of direct-to-consumer ancestry testing in South Africa, which is starting to boom. Online consent will be requested from potential participants via REDCap, in addition to the collection of incredibly dense phenotypic information. So the thought here is that we'll capture a great amount of genetic diversity within Southern Africa, particularly of populations that remain particularly quite understudied. So the goal here is to create a large-scale resource that facilitates population genetic and pharmacogenetic research based off of the unique phenotypic information that we're getting, which enables us to do pharmacogenetic analyses in a wide array of Southern African populations. So another area that we are interested in is looking at population-specific genetic recombination maps for Southern African populations. And we're doing this in 3 ways. So we're doing a linkage disequilibrium-based recombination map, a local ancestry inference-based recombination map, which I'm focusing on, and then a pedigree-based recombination map. Each have their own advantages and disadvantages, and we hope to harness all of these to generate a framework that is able to produce population-specific recombination maps for admixed populations. So another one of my students, PhD students, Mr. Gerald van Eerden, is particularly interested in pedigree-based, LD-based, and LD summary statistic-based methods to generate recombination maps. So he is looking at the correlations between these maps across different populations, and preliminary analysis, not surprising that the NAMA recombination map is the most distinct on a large scale. We're also looking at how the recombination map choice affects the placement of ancestry switch points in local ancestry inference, and how this can affect downstream analyses such as admixture mapping. For his PhD, he's looking at other factors that affect the placement of recombination hotspots, such as PRDM9 variants. And his ultimate goal is to look at how recombination, or how the population-specific recombination map can be used to improve local ancestry inference, and how that ancestry information can be most appropriately utilized in genome-wide association studies. So one of the other aspects, if you recall, was that genetic tools and computational algorithms, how accurate are they in capturing the genetic diversity in Southern Africa? So this was recently published in BMC Genetics, looking at how accurate the most widely used population genetic tools are for a 5-way admixed population. We use simulations to estimate the accuracy of these computational tools, admixture specifically, and arithmix. You can see from these correlation plots that that arithmix performs better when it comes to global ancestry estimation. It is also pretty robust if you give it misinformation, if you give arithmix misinformation. So the simulations were based off of a generation time of 15 generations. So we gave arithmix incorrect generation times of 10 and 20, and we looked at how accurate it was. And we were happy to see that in most cases, although it's a give and a take, in some instances, the 15 generation time proved to be pretty accurate. And we went ahead and looked at the accuracy of local ancestry inference, and we were pleased to see that we could achieve an accuracy of close to 90% for this complex 5-way admixed population. So as I said, this was recently published in BMC Genetics in collaboration with my two hosts. So linking this to infectious disease risk, particularly tuberculosis, what we are interested in is when it comes to TB as a disease, you need three factors. Obviously the pathogen, a conducive environment for disease, and a susceptible host. So we're interested in the susceptible host portion. So linking population structure, infectious disease risk, publishes a review reviewing prior literature. And it was very evident that although there have been a number of TB host genetic studies, a lot of these results remain unreplicated and the importance thereof remains unknown. So one of the first things that I did was to take all of these prior GWAS and perform a post-GWAS analysis to identify predicted regulatory variants and their association with tuberculosis susceptibility. And very interesting, we got some very interesting results from this. Another one of my students, PhD students, Yolande Swart, also took an alternative approach to standard GWAS, and she performed admixture mapping of TB susceptibility in a 5-way admix South African cohort. And she too identified some very interesting significant associations. And for her PhD, she's taking it a step further, and she's going to perform EQTL mapping to look at differentially expressed genes amongst the TB case cohort, a type 2 diabetes cohort, and a type 2 diabetes and TB co-infected cohort, to look at how type 2 diabetes may be related to TB. So future studies in this infectious disease realm, we are starting up a TB drug pharmacogenetic study. I've always been interested in natural selection, but that's in a highly complex admix population is incredibly difficult to perform those types of analyses. We've recently become involved in assessing the utility of polygenic risk scores in TB susceptibility, and based on the current climate, we have become involved in COVID-19 host genetics, and we've just received ethical approval to go ahead with that project, which is really interesting. So let's move a bit more to clinical genetics. So I'm involved in a master's project, Samantha Bailey. She is looking at a rare autosomal recessive condition called Mabry syndrome. It results in severe intellectual disability, increased levels of alkaline phosphatase, and the patients experience seizures. The specific patient was sent to Invitae for a specific panel for this disease or disease associated with this, and came up negative, and was decided that we were going to send the patient for whole exome sequencing. We found a novel homozygous variant in a known Mabry syndrome associated gene. In silico prediction determined this to be pathogenic and was Sanger confirmed. And we identified further individuals with this variant, Sanger confirmed, and we hypothesized that this could be potentially found effect in this group of patients, or this indigenous African population. So other diseases that we are interested in is lethal multiple pterygium syndrome. We have quite a large cohort, surprisingly. We suspect that there also might be a founder effect there. We are involved with a neurologist at Heidelberg to look at genetics of ALS. And then we have a primary immunodeficiency group within our division that has been quite successful in identification of novel variants associated with PID. So my last area that I'm interested in is bioethics and ethical, legal, social issues surrounding genomic database and databases and genomic research. So I currently sit on the HREC committee of the university, and I've obviously associated with that completed a good clinical practice course. But I'm very interested in receiving formal training in bioethics in 2021 and to incorporate research questions linked to bioethics and LC issues in some of our projects in the future. And I'm definitely open to potential collaborations and opportunities linked to bioethics or PopGen in general. So how does this benefit populations worldwide? So one of the biggest areas that would be affected is obviously the increased availability of genetic data for use as ancestral data in other populations, as all populations are linked. We would have an improved understanding of the origins of African populations, and with that, origins of other populations worldwide, as well as the optimization of computational algorithms and genetic tools for complex admixture scenarios, which is not only present in Africa, but worldwide. Without these people on this line, this research would not be possible. The funders, study participants, we thank you very much, and I am definitely open to any questions.
Thank you, Caitlin, for that amazing presentation. I, for one, am a bit new to the field of population genetics, but looking at your work, it has really inspired me. And let's see if there's some questions from the chat box. Just as a reminder, yeah, you can post the questions in the Q&A box. So we have a question from Lindley Darden, and the question is, what specific ethical questions are relevant to your work?
Thanks, Lindy, for that question. It's a very good question. I think ethical concerns for me is very wide-ranging. I think it starts off when you consent individuals into your research. How effective is your consent process? Do they understand the consent process? Then it would be that you perform your research in an ethical manner, taking into account your research participants. And then if you take it, to the next step and you perform the research and you have your research results, how do you disseminate those results to your participants? That's something I'm particularly interested in. And then again, as well, we've had particular experiences with how do you compensate research participants for their time, inputs, and expenses associated with taking part in research. And then if you look at genetic databases and in terms of confidentiality, I mean, being anonymous is in this day and age is unfortunately not possible. So, you would need to be pseudo-anonymized, various ethical implications there as well. So without me, I can talk about half an hour on this, but there are various questions that I'd like to answer. It would just be picking and choosing what is possible given the current circumstances.
Thank you. Thank you, Caitlin. Any other questions? Please, you can post in the Q&A box. Okay, I just have a question from my side, Caitlin. From the analysis from the comparison of the different tools that you made for the admixture, would you recommend that we need to sort of build more pipelines that are specific for African populations?
Yes, I mean, from the analyses that I did, I mean, I have collaborators who assisted on the design of RFMix, and it was very evident to me that RFMix is better suited for global and local for global ancestry, and obviously for it's accurate for local ancestry. And I think in this day and age, you know, things are becoming more and more adaptable and more applicable. But I think the awareness just needs to be there that you as a bioinformatician, you shouldn't be creating tools that cannot be used on all populations worldwide. And particularly in our case, we struggle with the extensive admixture that we have in populations in South Africa, and which is unique. But I think, as I said, as bioinformaticians, you need to be recognizant of the fact that there is that extensive admixture out there.
Okay, thank you. And then there is Kevin Eso, who is saying, thank you for the presentation. Given your experience in analyzing population genetic data of South African individuals and others, would you say there is a breakdown between anthropology, culture, and genetics, or our anthropological and cultural classification of human populations is consistent with the genetics?
Very interesting question. And my first paper in genetics, I think, did discuss that briefly. I think there is definitely some breakdown between anthropology, culture, and genetics, or how you would use those fields to classify human populations. And I think it's quite a tricky subject, because in each field you have specific data that is used to make conclusions that are very different in themselves. So I think we should use them in a complementary manner, rather than saying that anthropology concluded X and genomics concluded Y. We should use anthropology, genetics, cultural studies to create a conclusion and harness the advantages of each.
Thanks, Caitlin. Just as a follow-on question, Prof. Scott Hazelhurst is asking that are there not huge risks in this regarding to reinforcing ethnic labels given history of colonization? And he's suggesting that we need to be careful of why we want to classify people. Nobody can comment on that.
Thanks, Prof, for that question. I agree 100% with your comments, and there are great risks. I think one of the things we have always done, and try to do even more so, is be recognizant of that fact. I think that one of the things that I perhaps didn't mention is that we don't classify people into groups based on genetic data. It's grouped on self-reported ethnicity that's collected, and we obviously don't reassign individuals or anything like that. But I agree, there needs to be you need to keep that in mind throughout your population genetics work, how you disseminate results, so as to not reinforce, as you say, the ethnic labels given during colonization. And we also adapt all of our research results and the way in which we perform research to the current climate in which population genetics is performed.
Okay, great, thank you. And then we have a question from Ronald Rodriguez Santiago, who is asking, do you encounter concerns from participants on how their data will be used? And he's saying that there are cases of companies reusing ancestry data from consumers for criminal cases and other purposes on the rise. How would you think these concerns can be addressed?
Thank you very much for that question. Yes, we have in all of our research had concerns from participants on how the data will be used. I think to answer your question is education. You really need to make sure that, well, education and informed consent, you really need to make sure that your research participant understands the research that is going to take place, that they are consenting to. You also need to have strict measures in place in terms of data security so that their data is not breached and that they understand that their data is being stored in a manner that would prevent a data breach. In terms of cases of companies reusing ancestry data from consumers for criminal cases, so I think you might be referring to the GEDmatch scenario, and in that case, the genetic data is uploaded to GEDmatch by the clients who have performed the direct consumer testing, so we wouldn't have anything to do with that, but that, as well as the recent data breach at GEDmatch, once again exemplifies the concerns for how their data will be used. And once again, it comes back to education and understanding how your data will be used. So I hope that answers that question.
Yeah, thank you, Caitlin. And then another question here from Darin Takemoto. He's asking, how will the phenotypic data be collected for the South African Human Genetics Database? And broadly, what types of data will be collected?
Thanks very much for that question. And it's a very good one. So given the current climate with COVID, it was a bit of an opportunity for me to think out of the box for phenotypic data collection. So what we've decided is to gather phenotypic data sort of at the basal level of height, weight, what else? Obviously age and gender, and then we look at whether the research participants was diagnosed with chronic diseases. So this is all self-reported, whether the individual has been diagnosed with a chronic disease, what medication that they take for that disease, as well as whether they have had any adverse reaction to that medication. Now we see this as in terms of the adverse drug reaction section, we see it as a bit of a pilot project to see what data we're going to get out, and obviously amend the phenotypic data collection accordingly. But this will also serve as a pool of data or control data for the population structure work, but also for some of the other analyses that we'll be doing.
Yes, thank you, Caitlin. I think just one more final question.
Sure.
This is from Maria. She's asking, which institution will be hosting the South African Human Genetics Database, and will it be accessible for research purposes?
So it'll be housed at Stellenbosch University, and it will be accessible for research purposes. the goal is to have it alongside the relevant publications, and it'll be uploaded to a archiving facility such as EGA that you'll need to undergo an application procedure, but as long as it falls underneath the purview of the informed consent, researchers will have access to it.